Niels, thanks for the kind introduction. So as Niels mentioned, I'm coming to you from a field of neuroscience called Connectomics. And today I'm very excited to talk to you about some of our methods that we use from machine learning and interactive data visualization to better interpret these really, really difficult and intricate 3D maps of the brain. So for those of you who have maybe not heard about the term connectomics, it's the science of creating and analyzing synapse-level wiring diagrams of an organism's nervous system. And what you're seeing here on the screen is like a big deal. Last year, the first or the largest whole brain connectome that was ever created came out in Nature. And this shows you every single neuron and every single synapse that is present in the brain of a fruit fly. So you can see now here, this is part of the central brain, and on the outside of the brain, you can see the optic lobes. So those are the neurons that are right under the eye. And these kind of maps really help researchers to kind of like better understand the hardware of the brain, like really the synaptic wiring architecture. And what's neat about that is that now in neuroscientific analyses, neuroscientists can really use these synapse level maps to, as a starting point for their circuit analyses. So an example here that you're seeing is the auditory circuit that was reconstructed in the brain of a fruit fly. So those are all the neurons that process auditory information. And analyzing the hardware of that circuit is really gives a good insight about how the function of the circuit works. And people are not only doing that for small model organisms like the fruit fly, but also for small parts of the human brain. So for example, here I'm showing you a slice from the HO1 data set that is about 1 cubic millimeter of human cortex. And just to give you a sense of the complexity of that data, here I show one of the 57,000 cells in that data set. And in green, I'm showing the all the incoming axons to that neuron in pink. So you can really, appreciate with that picture how dense that graph is. So you have like one node, the neuron in pink, and it has like all these like edges that are coming to it through those green axons. And I want to talk about two projects briefly that I've been doing in this space. But before that, I will briefly introduce the Connectomics pipeline. So usually researchers start with extracting a small piece of brain tissue, either from a human or a fly. And as a next step, they like cut it into very, very thin slices, usually a couple of nanometers thick. And after those slices are cut, they're imaged using typically an electron microscope at about like 8 by 8 nanometers resolution. And more recently, people have also been using expansion microscopy and light microscopic approaches to acquire those data sets, which is very exciting. And then as a next step, we use computer vision algorithms to sort of like align those slices into a 3D tissue volume. Once that's done, we use 3D convolutional neural networks to segment the data, so attach labels to each voxel in the volumetric representation of the tissue. And after segmentation is done, we use a step called proofreading, which essentially is required to correct and identify reconstruction errors that are happening in the previous segmentation step. And once that's done, and we're like certain that the data has a certain quality threshold and is accurately proofread, we can actually analyze the resulting connectome network. And in the first part of my talk, I want to briefly explain some of the challenges and approaches that we've been developing about connectome proofreading. So it turns out proofreading is a major bottleneck in connectomics. And an example is shown here where if you're familiar with connectome data, you can very easily tell that this rendering here is not only one neuron, but actually two, but they have been incorrectly reconstructed and have been accidentally merged together in the segmentation step. So what we have to do right now, and that's currently the state of the art, is that a human proofreader goes through the data set and manually identifies and corrects those errors. And once that's done and this merge error has been fixed, it looks somewhat like this, where we have now a red neuron and a black neuron that are correctly split apart. And again, to give you a sense of the scale at which this has to happen in modern connectomics, depending on the level of detail that a human proofreader works, it can take between 1 to 10 hours to proofread a single neuron. And with my extremely optimistic guess, which many people in connectomics probably would not share and say that's too optimistic, it would take about 27 years of work for a single human to proofread one single state-of-the-art large-scale connectome, such as the HL1 dataset. So right now, how we're tackling this is through crowdsourced proofreading. So we have with others developed and deployed these interactive proofreading platforms. So here for the HL1 dataset, a human proofreader is going in and by marking the two different neurons here with those points, they can interactively split those reconstruction errors apart and correct certain mistakes. And this process by now is fairly optimized. Like it's all web-based. A lot of users can work at the same data source. So there exist systems like the one mentioned here at the bottom called Cave that you can think of as some sort of like Git version control for connectomics. So like many users can proofread and update those connectomes interactively. But still, even with that very optimized process, recently a report came out from the Wellcome Trust. That's a funding agency that supports connectomics research. And they've been trying to estimate what it would cost to, create the next large-scale connectome, the next frontier in connectomics, which is like a whole mouse brain connectome, is orders of magnitude larger than what we currently can do. And in that report, they've been saying that, okay, like one of the largest bottlenecks actually is proofreading such a mouse brain connectome. And they estimated it to cost between $7 and $21 billion just to proofread that connectome. And obviously nobody's gonna pay that. So $21 billion right now is infeasible. So we need to significantly cut down on that cost. And to that end, we've been thinking about whether we can learn certain machine learning models that automatically proofread those next generation connectomes. And now that we have all these like expert annotations from previous projects, we can use those as training data. And so, for example, what you're seeing here is a point cloud that was sampled from an incorrectly reconstructed neuron. So there are multiple neurons in there. And in our preprint that is mentioned here at the bottom, we developed a machine learning model called the point affinity transformer. And using that point affinity transformer, we can give this whole point cloud as a context to the model and then ask for a pair of points that we can randomly pick from that point cloud. Hey, like how likely is it that those two randomly picked points belong to the same neuron or not? So the model predicts some sort of affinity score. And that affinity score is 1 if those 2 points belong to the same neuron. It's 0 if they don't belong to the same neuron. And if we do that across all possible combinations of points in that point cloud, we get something called a point affinity matrix, what you're seeing here. And if you think about it, you can sort of view this as a point distance metric. So for each pair of points, there exists a pixel in that matrix, and that pixel tells you how likely is it that those 2 points belong to the same neuron. And now that we have this distance metric and this point cloud, we can actually just use conventional clustering algorithms to sort of cluster this point cloud based on that distance metric. And using these clustering algorithms, we can derive new updated labels that sort of like carve out the individual neurons out of this incorrectly merged one, which is a very promising approach to sort of speed up the next level of proofreading work in connectomics. Now I'm gonna shift gears a little bit and transition to what we've done in working with connectomes once they're proofread. So moving into the analysis part of the graph structure that has been reconstructed. So it's not trivial and like how to interpret these data sets, and a naive way maybe would be to just plot a big node link diagram of a connectome, and then you end up with something like this, which is a crazy hairball and nobody sees anything. And if you zoom in a little bit, you can see the directed graph structure of the connectome. And it turns out that analyzing connectivity motifs is actually a very promising way of looking at these data sets. So a motif would be like a small subgraph. You can think of it as some sort of a fundamental atomic compute function that repeats itself over and over again in the network. And so in this small subgraph, we can see this example motif, this feed-forward network, occur about like 2 times. And this is a very common approach for neuroscientific analysis. So some of my colleagues at Janelia have, and many more people have published papers that essentially identify certain network motifs, for example, in the Drosophila connectome, that allow it to, in this case, perform flexible navigation and context-dependent action selection tasks. And to this end, we've developed an interactive visualization analysis tool that allows, it's called VMO, that allows to interactively search and analyze such network motifs. So here's a quick demo. A user is drawing a 4-neuron motif and connecting it through directed edges. And the users can also additionally set certain constraints on that motif. So for example, here the user is setting like a specific brain region, or here a specific connectivity strength. So about 20 synapses need to be between A and B. And then they can search that connectome for instances, so neurons that implement that sketched pattern. But that's actually not the full story. So what we quickly realized is that it's very critical to correlate essentially the network connectivity with the neuron morphology. And what I mean by that will become very clear when you look at this picture. So we know that the neurons that are shown on the right implement the connectivity pattern on the left, but at least to me, it's very unclear, like, how is this pattern, integrated there? And it's really important to understand how this graph structure maps to the 3D morphology of those neurons. So in order to tackle that, we've been building an interactive abstraction technique where once a user has sketched and found a particular connectivity motif, they can render it in 3D here. And then by dragging the slider at the bottom, they can first like prune away all branches of the neurons that are not part of that connectivity motif. And then once they drag the slider a little bit further, the neurons getting spatially pulled apart, and we draw links between the pre and postsynaptic sites. And that makes it much more visible how this like triangular pattern is like implemented in those 3 neurons. That's like one way of better mapping from the graph topology to the neuron morphology. And so we've been thinking on how to further extend this, and really the inspiration that we're drawing from here is from some findings from the HR1 paper, where our collaborators have found these multisynaptic connections of 2 neurons, where the synaptic connections are really, really close by on the neuron branches. And our collaborators are hypothesizing that these synaptic connections are so strong in such a proximal way due to learning. However, if you just represent this connectome as a conventional graph, the graph motif would look somewhat like this here, which is kind of ambiguous and is not really capturing the structure that we are looking for. It could be that those synapses here shown in red are electrically really like far apart, like in this example, or that they're really close together, which is more like what we're looking for in this example. However, like the graph motif is still the same. It's not very specific in this case. So we're thinking about how can we build more spatially aware graph data structures and integrate them into our interactive analysis pipelines in order to reflect these graph motifs better. And now I want to acknowledge my amazing collaborators, in particular my advisor Hans-Peter Pfister and my other mentors Johanna Bayer and Srini Turaga at HHMI Janelia, and also all my other amazing co-authors and people that really support me with all this work. I also outlined all the publications that I touched on in this talk, and I'm now very excited to hear your questions.
First of all, perfect timing. Thank you, Jakob. Okay, are there questions?